This is the concurrent session entitled Subcellular Correlates of Consciousness. We have 7 speakers, 4 who are with us today and 3 who will be joining us remotely. Our time is going to be a factor. I will try to keep things pretty much tight on schedule. And we'll begin with our first speaker, Matt Mihelic from the University of Tennessee. And his topic is quantum DNA, modeling, evidence, and implications.
Hello, I'm Matt Mihelic. I want to tell you about how DNA is a quantum logic processor. I'm going to tell you about it, how to model it, the evidence that we have behind it, and then some implications of that. This is not a new concept. Schrödinger, back in 1944, in his book, What is Life?, said that adaptation is necessary for life and that quantum jumps between energy states are necessary for adaptation to occur. He said that only a crystal can provide the stability necessary for such a quantum system, but a regular repeating crystal doesn't have enough information to support life. So whatever that information was in the nucleus of the cell, it had to be some sort of an aperiodic crystal. Very much influenced by Schrödinger were Watson and Crick, who, using X-ray crystallographic data and insights supplied to them by Franklin in 1953, figured out the structure of the DNA double helix. So they had deduced the structure of the quantum mechanical system that Schrödinger had intuited. And what we're going to talk about today is the function of that system. 3 things coming together that we're going to talk about to make DNA a quantum logic processor. The first is coherent conduction of electron spin states, then spin filtering of electron spin states, and finally a quantum gate in which those spin states can interact. This is a 3D model of the DNA molecule, and you'll remember that on the inside in blue there, you can see that the nucleotide base pairs are stacked like pancakes. Now they're aromatic molecules, so their p electron clouds above and below the molecule will run together and delocalize electron longitudinally along the DNA molecule. Electrons can thereby be coherently conducted along this pi stacking interaction of these aromatic nucleotide bases. Now, this depends in part on the base pair sequence and in part on the base pair relative geometry. So you can't think of DNA as a great big long quantum wire, because it isn't. When you look at DNA under a microscope in a living cell, it's always kind of jiggling around. It's always moving. So that geometry is always changing. So we need to think more in terms of segments of coherence that are dynamically shifting and overlapping as the geometry changes. Now, at the same time, electrons moving longitudinally along the DNA molecule are subject to a very efficient electron spin filtering effect, as the spin of the electron interacts with the helicity of the DNA molecule. Goller and his colleagues took electrons and shot them longitudinally along the DNA molecule. They found that spin down electrons went all the way through, but spin up electrons got filtered out. This is a distance-dependent effect. The electron had to travel far enough along the helix, the double helix, in order for a spin motive force to develop on that electron and kick it out. So in DNA, you have 2 simultaneous processes where electrons are being coherently conducted along a very efficient spin filter. But coherently conducted and spin filtered to where or for what purpose? Well, these 2 simultaneous processes can theoretically provide the means by which an individual electron or its spin state can be deposited or read into a specific nucleotide and thereby affect the deoxyribose moiety of that nucleotide. In red here, we see the one of the deoxyribose moieties. Now, here's a close-up of that Furanose ring of ribose or deoxyribose. It's a 5-carbon sugar. Carbon 1 is connected to the nucleotide base. Carbons 3, 4, and 5 are part of the phosphate backbone, and carbons 1 and 4 are connected together by an oxygen atom. It's not a stiff molecule because of that. It's a floppy molecule. It flops around in what they call a pseudo-rotation. And there's various conformations around that pseudo-rotation. The important ones that we're going to look at today are the C2 endo and C3 endo conformations that are separated by an intermediate O1 endo conformation. This is a plot of the deformation energies around that pseudo-rotation. You'll note that C2 endo and C3 endo occupy the lowest points of this deformation energy. They're separated by a relatively low energy barrier of 0.6 kilocalories per mole. This creates a double well potential separating what is an enantiomeric symmetry. C2 endo and C3 endo are chiral enantiomers. They are stereoisomers. What's the ultimate determiner of chirality? The ultimate determiner of chirality is electron spin direction via spin-orbit coupling at a chiral center. And in the deoxyribose moieties of the DNA molecule, the covalent bond between the C2 and C3 carbons share electrons between two chiral centers. So theoretically, as an electron or spin state is being read into the deoxyribose, its spin direction can determine an enantiomeric selection as its orbital angular momentum can overcome or randomize that double well potential. This would constitute a symmetry break. What's interesting here is it is a symmetry break that occurs across an energy barrier very appropriate to the Landauer limit, okay? That 0.6 kilocalories per mole is just a skosh more than what Landauer described as the quantum limit of a deterministic bit. If you have less than KT natural log 2, which is 0.0178 electron volts per bit, if you have an energy barrier of less than that, separating a double well potential, you cannot have a deterministic bit. All you can have is a statistical relationship. And because of this energy barrier and this flip-flop between these C2 endo and C3 endo, that would have a quantum vibration of about 4.3 terahertz. Now, remember that there's an deoxyribose moiety on both sides of the base pair. And if the deoxyribose moieties on both sides of the dinucleotide simultaneously shift from C2 endo to C3 endo, that base pair is going to roll by 22 degrees. And this is going to change the geometry of that pi stacking interaction. You can see if you roll one of those base pairs by 22 degrees, you're going to break that coherent conduction along the DNA molecule. So the deoxyribose moieties are strategically situated between the coherently conducting quantum information of the nucleotide base pair sequence and the deterministic classical information of the topological structure of the phosphate backbone, and thereby can mediate the quantum to classical transition of information across a deterministic energy barrier that is appropriate to the Landauer limit. So is this quantum to classical transition across the deoxyribose enantiomeric symmetry the quantum measurement that provides a decoherence for the observer effect? Well, Heisenberg said that the transition from the possible to the actual is a necessary component of the interpretation of quantum theory. And von Neumann said that somewhere between the physical and the psychological, there's a Schnitt, which is a German word for cut. There's a Schnitt that occurs between the physical and the psychological. Penrose and Hameroff say that consciousness emerges from quantum decoherences at sites of proto-consciousness. And Landauer would say that hey, if you're going to have this quantum to classical transition, it has to be across a deterministic energy barrier of KT natural log 2. So this helps us to understand the Bohr versus Einstein controversy, where Bohr said everything's statistical, and Einstein said, well, some things have to be deterministic. Great theory. How are you going to show it? Well, we wanted to validate this. We were inspired by the work of Rita Pizzi. We felt that entangled electrons can be separated into separate DNA strands during replication of the DNA, and thus into separate cells and into separate cell cultures. We wanted to show non-locality between separate cell cultures because non-locality is the hallmark of quantum logic. So we took a culture of rat neuronal stem cells and we grew it for a week. And then we split that culture into two separate cultures that we grew for 3 more weeks on little microelectrode array culture dishes that have little microelectrodes on the bottom where you can monitor individual cells. And then we monitored the cells while we hit cell culture A with a laser. And we found that laser stimulation of culture A caused depolarizations in A, but it also caused correlated depolarizations in B. I want to show you a little bit about what our data looked like, but I have to explain what you're looking at here first. Our equipment could monitor 8 data channels at one time. So we dedicated 3 data channels to look at cells in culture A, 4 looking at cellular depolarizations in culture B, and 1 to keep track of the laser pulse timing. We monitored each iteration here for 5 seconds. We pulsed a 650 nanometer laser, which is essentially a red laser pointer, for 50 milliseconds every half a second. So we got 10 pulsations. And what you're seeing in these cells in this example are just random depolarizations of cells. And we did see plenty of random depolarizations, but we saw other things too. You'll notice in the top cell here in culture A that this cell went just by virtue of the fact that it was being hit with the laser in this pattern, had gone into a series of depolarizations, oscillatory depolarizations running at about 130 hertz. And you'll notice also that cells in culture B were doing the same thing. But remember, the laser's only hitting culture A. Occasionally, we were fortunate enough to see the initiation of these oscillatory depolarizations. And when we saw them, they always happened on or immediately after the 4th laser pulse. We saw some other things too. In this iteration, you'll see that all the cells in culture A are in these oscillatory depolarizations. None of the cells in culture B are in these oscillatory depolarizations. However, you will note that this cell in culture B is responding to the laser pulsations, but it started on the 4th depolarization. Correction, it started on the 4th laser pulse. But we had a phase 2 of this also. In phase 2, we exposed culture A to an anesthetic gas, theoretically to interrupt the function of the microtubules. But culture B, we theorized, would still respond to the laser stimulation of culture A. Penrose and Hameroff theorized that microtubules are a source of quantum effects in biological systems. And microtubules can thus be reasonably modeled as quantum wires that provide locally coherent quantum communication to and from the nucleus. Microtubules do a whole lot more than just communicate. They also will store information and process information as well. But we felt that we could interrupt the quantum mechanical processes of the microtubules with a general anesthetic gas. We used isoflurane to theoretically isolate the nucleus. And thus, any non-local communication between cells that would continue in the face of this anesthetic gas would be attributable to what's in the nucleus, which is essentially the DNA. So in this iteration, we have anesthetic gas going to culture A, and we have laser hitting culture A in the same pattern. And you'll notice that in culture B now, instead of seeing an initiation of these oscillatory depolarizations, we're now seeing a termination of the oscillatory depolarizations. Never did we see initiation of oscillatory depolarizations in phase 2. Never did we see termination in phase 1. We wanted to look at some correlations here. We wanted to see if this cell I can't get my pointer to work. There it is. If we want to see if this cell in culture A, or see how correlated these cells were. And we wanted to look at this time period between the 1st and 2nd laser pulse when everything seemed to be phase locked together, and in this second period of time between pulses, laser pulses 8 and 9, when all this oscillatory depolarizations were turned off. And you'll note in this, and so we did Spearman-Rowe correlations. And when you look at this cell in culture A compared to this cell in culture B for this time period, there's a 0.8 correlation, which is very, very high. It shows that essentially these cells are phase locking. But in this second line, this cell in culture A and this cell in culture B have no significant correlation. So not all the cells are phase locking together. If it did, you know, in your brain, it'd be a seizure. So it's probably a bad idea. But then when you look at cells in the same culture, these two cells had, these three cells were not correlated. Now we go over here to this second segment where all these phase lockings, I guess, are turned off. And now this cell in culture A and this cell in culture B have no significant correlation, still no significant correlation between these other two cells in separate cultures. However, when you look at the cells in the same culture, now you have a 0.4 correlation, which is a relatively good correlation. What are the implications here of these experimental results? Well, the experimental hypotheses were confirmed in that non-local communication was demonstrated between cells in separated cell cultures, and it persisted in the face of the general anesthetic. Such non-locality indicates entanglement, which is the hallmark of a quantum logic system. So DNA is a quantum computer, quantum logic processor. The non-local communication between cells was modulated by laser pulsations and by pharmacological manipulation, so we can control it. These correlations that were experimentally observed were not due to interference from an electromagnetic field. The depolarizations are biological phenomena that continue between the laser pulses. Not every cell exhibited oscillatory depolarization, as you would expect if there was some kind of electromagnetic interference. When non-local initiation of phase lock was observed in culture B, it always occurred on or immediately after the fourth laser pulse, indicating very efficient and rapid calculation. If this was interference, you'd see it with every laser pulse. And finally, a definite non-local pharmacological effect was demonstrated. That's pharmacology, that's not electromagnetic. So this has broad implications for consciousness studies. This is, you know, one of the possible schnitz of coherence. Pardon me? Von Neumann, yeah, what a von Neumann schnitz. But in quantum computing, you know, DNA is a quantum computer, and we feel like with terahertz speed microprocessors, we can emulate it, or we can interface with it. As far as artificial intelligence goes, yeah, you know, this kind of throws a little bit of a monkey wrench into the idea that neurons are just integrate and fire complicated transistors. For system science, what we think of as complex adaptive systems are in reality quantum adaptive systems. And that means a lot for social sciences. In the biomedical sciences, you know, neuroscience, genetics, et cetera. I mean, the cell biology, a lot of potentials there. So that's my quick introduction to this. These are some related publications of mine. I'll be glad to entertain any questions, or if anyone wants to send me an email, that's my email address there. Okay.
Thank you, Dr. Mihelic. Did I pronounce it right?
Yes.
I think we have time for a few questions now, and I didn't pick up any online. I'll leave it to Matt to let us know if there are any.
One there. Yes, sir.
So in your analysis of the correlations between the signals in sample A and sample B, did you look at the distribution of those, of the correlates, as opposed to just it looked like there's only six samples or so on each side. Did you look at the distribution of correlates across a larger sample size?
No, we did not. We, we had limited access to a statistician, but what we found we thought was extremely important.
Yeah. Do you still have the data?
Yes.
Hi, my name's Mike. Thank you for the talk. You mentioned potentially interfacing it as like a stepping towards the future possibility. Have you ever thought of different ways you might consider interfacing?
Yes, absolutely. After we came up with, after this data came available, I hooked up with some guys formerly from Silicon Valley, and they have the ideas and now the intellectual properties on a terahertz speed microprocessor that works on nano vacuum tubes and lossless transmission lines. And it can run at terahertz speed across the chip. And we feel like this will eventually be able to interface with DNA and or emulate it.
Awesome, thank you.
Thank you.
I think we have time for one more question from me.
Yes, sir.
Since this phenomenon occurs in DNA, is it subject to evolution?
Well, this is, in my opinion, this is the mechanism of adaptation. Evolution and adaptation are often intermingled as far as that goes. So, and indeed, this was the concept that Schrodinger was discussing in What is Life in 1944. So it is certainly the mechanism of adaptation from my point of view.
Okay. Thank you once more. And I think we're ready for our second speaker. Am I pronouncing it right? Amal Alachkar from UC Irvine. And her talk will be about Cilia, from vestigial organelles to vital players in high-order cognitive functions.
Hello, everyone. Thank you for staying until this session. And okay. So we listened today afternoon to 4 talks about microtubules, and like in this conference, really we enjoyed listening to more talks about microtubules. So Cilia is plural, is one of those organelles that are microtubules based. So this is the structure of cilia. It's, well, they are organelles that are microtubule based, and they work as cell antenna. And as you see, the 9 doublets of microtubules, and they have exactly the structure of the cytoskeleton microtubules. There are 2 types of cilia: the motile cilia, in which there are a central doublet of microtubules, while the primary cilia, which I will speak about today, they have only 9. They are called 9 plus 0 because they don't have central doublet of microtubules. So cilia are ancient as life on Earth, and Paramecium, which is unicellular organism, had cilia, and the Paramecium used cilia for swimming purposes. However, we know that human brain also has cilia. And there are 2 types of cilia indeed in the brain, in the primate, in all animals indeed. Almost all cell types in all organisms have cilia. So in our brain and in animal brains, there are the motile cilia, which are around the ventricles. They move the CSF, the cerebrospinal fluid, and there are the primary cilia, which really exist on almost all neurons and glial cells, such as astrocytes. So these are brain sections. I hope you can see the cilia. Yeah. So those are brain sections of mouse brain from my lab. And as you see here, indeed, all brain regions contain cilia. And even like you see here, this is the hippocampus. You see that astrocytes can express cilia, but also neurons express cilia. So this is prefrontal cortex and the striatum and nucleus accumbens. We were motivated to do this research on cilia based on that the majority of ciliopathy ciliopathies that are studied on humans are associated with cognitive dysfunctions. But it's interesting that for 100 years, the functions of cilia in the brain was totally dismissed. So only a few decades, they start to speak about the role of cilia in the kidney, in the cardiovascular system, in the respiratory. But brain, we still know very, very little about what cilia can do in the brain. So for us, like, okay, so we started with correlational studies. So the closest disorder to the cognitive dysfunctions is schizophrenia. And so we looked at database of all the transcriptomes, the 28 genes, and we found that, of course, that's in collaboration with Dr. Pierre Baldi in University of California, Irvine, found that around 50% of cilia transcriptomes are dysregulated in schizophrenia. And of course, the database include post-mortem prefrontal cortex from patients with schizophrenia. And as you see here, there are like many that are upregulated genes and downregulated genes. It's interesting that cilia have G protein coupled receptors, which are important for all neurotransmitter systems in the brain. And it's interesting, about like 20 G protein coupled receptors exist on cilia. Out of those 20, around 15 GPCR, G protein coupled receptors, are dysregulated in schizophrenia. And we know that G protein coupled receptors are a target for a lot of treatments in psychiatric disorders, particularly in schizophrenia and, for example, dopamine receptors. And we see that indeed the dopamine receptor 5, dopamine receptor 2, which are proved to exist also in cilia. Of course, beside the cell membrane, they are dysregulated in schizophrenia. So this is only correlational evidence showing that, okay, so there might be a link between cilia dysfunction and mental disorders. But then we thought, okay, are there conditions, like just like within the normal conditions that cilia are dynamic? And we looked at the transcriptomes, like the human lifespan transcriptome, and we found that at least 67% of human brain cilia transcriptome follow linear regression model. And that even we didn't use other like nonlinear regressions. And the majority of cilia genes that show change with age, indeed, they show increase with age, while the less showed decrease with age. And as you see here, the ventrolateral parietal cortex showed the highest number of dynamic cilia genes. That means like this region of the brain showed the most cilia genes that change with age. Then we looked at, okay, so we have cilia genes are dynamic across the lifespan. What about during 24 hours? And we were very, very surprised that in the majority of the brain regions, and this study, we used the database of baboon brain that was published in Science in 2018. We took the data, the raw data, and we analyzed, of course, like Dr. Baldi's lab analyzed the data. And we found that in many regions, in at least 12 regions of baboon brain regions, the cilia transcriptomes show higher circadian rhythm than any other genes in these regions. But it's interesting that the peak phases of cilia transcriptome in different regions look different. That means really cilia in different regions of the brain do different functions. And that even motivated us to look at different functions. Not only that, we found that the cilia substructural organizations, that the genes in these substructural organizations of cilia have different peak phase. For example, while in the kinesin, there are different genes that are part of the kinesin system, they have peak during the day at around like 7 hours, while, for example, the intraflagella B and A, they have peak at 15 hours. So then we said, okay, so can we really manipulate the cilia structure? All these about the gene, the transcriptomes of cilia in humans and baboon. Can we really manipulate by manipulating some neurotransmitter system? So in my lab, we are interested in melanin concentrating hormone because we found that this system has been associated with schizophrenia, but also because melanin concentrating hormone receptors exist on cilia. And in collaboration with my collaborator in Japan, Dr. Yumiko Saito, so she took brain sections of rats and she applied melanin concentrating hormone directly. And then she found that the cilia length really decreased. And we thought like, okay, so that's in ex vivo. What happens if we manipulate the system in vivo? So we used several ways to manipulate melanin concentrating hormone. We activated the system either pharmacologically by injecting the neurotransmitter itself, the peptide melanin concentrating hormone, or by optogenetic methods or chemogenetic methods by those like DREAD receptors. So what we found that, as you see here, there is shift in the length of cilia. So activation of the melanin concentrating hormone in the brain of mice causes decreased cilia length. On the other hand, we said, okay, so what happens if we inactivate the system? So by either antagonist of the receptors or by removing, like having knockout mice of the receptors or knockout of the peptide itself, we found the opposite. Cilia length really increase when we inactivate the system, either pharmacologically or genetically. So but then what's the implication of cilia and its length in the cognitive function? So to answer this question, so now we want to really look at the causal effect evidence. So what we did here, we removed cilia genetically from the striatum by using the Cre-Lox technology. And we know that IFT88 is a gene that's very, very essential for the cilia genesis. So we injected Cre to cut, it's like it works like scissor, to cut this gene from a specific area, which is the striatum, which is very much enriched in cilia. And as you see here, when we removed IFT88, we removed cilia. I hope it's clear the difference between those two pictures. And then we challenged mice in different behavioral assays. So first, we just like throw them to look at spontaneous motor activity. What we found, they are fine. They look normal compared to the control animals. However, when we challenge the animals, the mice in something called rotarod, that they need really to learn different skills in movement, because we rotate this machine and we increase the speed of rotation, they start to show deficits. That means when they are challenged with something that they need to coordinate and to look at the timing of the movement and to learn new skills, they failed. Then there is another essay that's very important, and indeed it is used for schizophrenia, because schizophrenia patients show deficits in this essay. It's pre-pulse inhibition. And in this essay, when we are exposed to pulse, we show a response. However, when are exposed to pre-pulse that's weaker than the pulse, we start to show inhibition, or like our response decreases. However, in schizophrenia patients, that doesn't happen. So in our experiments, we expose the mice, we put them in chamber, and we expose them to loud sound is like 120 decibel, then they startle. Then when we expose them to pre-pulse, that like 68, 71 decibel, 77, they start to lose their response, startle response. So what we found that animals that lost cilia from the striatum, they have deficits. So no matter how much like we expose them to pre-pulse, they keep responding to with startle to the high pulse. What we found also that these animals that lost cilia show increased grooming behavior. That's repetitive behavior. Again, those two experiments show that really those animals have problem with responding to the environment and just like adjusting their timing behavior. So we moved to higher level of behaviors, working at memories. And in very simple experiment, this is working spatial memory assay. And so just like T-maze, we throw the animals and we look how many mistakes they do when they navigate the space. That's why it is spatial memory. And we found that the animals that lost cilia, really they have deficit. They do a lot of mistakes in alternating between the right and left arm. Not only that, they take much longer time to decide where to go. So they have decision making like longer latency to make a decision. However, it's interesting that when we challenge these mice in long-term memories, they showed just normal responses. For example, there's this novel object memory. We have two objects identical, and we train them to just like explore them. And after 24 hours, we remove one of them, and then we put a novel object. And we see that both the control and the cilia knockout animals show more tendency to spend time with the novel object than the old object. Similarly, in spatial memory, that's long-term memory, we have two objects, identical object in the first day. Then after 24 hours, we move one of them to different site, and control animals spend more time with the novel location, and cilia knockout also do the same. The last experiment, which is the most stressful one, is the contextual memory, in which we shock the animal. We expose the animal within a context. So we put them in chamber with a context like striped walls or something that they recognize the place. And then we do foot shock. And then the response is freezing. Then we take the animal to their cages. We bring them the second day. We put them in the chamber without exposing them to any stress or any foot shock, but they freeze. And we see that both the control and the cilia freeze after 24 hours. That means they remember the context of the shocking. So that means really, I mean, these animals that lost cilia from the striatum have normal long-term memory. So knowing that the striatum really is a hub that receives projection from several regions of the brain, particularly different regions of the cortex, and at the same time, it sends executive actions through like this circuit of the basal ganglia. So we according to our data, the first conclusion is that striatal cilia may play an important role in acquisition and brief storage of information. And of course, that include learning of new motor skills and executive actions. However, there is no role for cilia and the striatum in long-term consolidation of information or maintaining habitual or learned motor skills. The second conclusion is that there's really causal effect of cilia on time perception. We know that according to our data, indeed, one aspect of all those disrupted functions like spatial working memory, but not spatial long-term memory, is like motor coordination, the repetitive behavior, all these have one aspect that's time perception or judgment. And that's why we believe that cilia might be involved in time perception, knowing that time perception is subjective experience and it is a fundamental element of human consciousness. That's why studying cilia might be very interesting as a neural correlate of consciousness. And in our model, we propose the cilia and the striatum as being involved in the brain clock function by maintaining the proper timing perception. And just like a note that schizophrenia patients indeed have deficits in time perception. And as a summary, so cilia, which are microtubules, they are dynamic systems. They are dysregulated in disorders of consciousness, such as schizophrenia. And their structure can be malleable, can be manipulated by some transmission systems. Cilia may serve high cognitive functions, and that's what we try to prove. And particularly, cilia may serve as a host for time perception. And that's why studying cilia might be very interesting as neural correlate of consciousness. And of course, now currently we are continuing to study the cilia in the role of cilia in different sides of the brain. For example, now our recent data show that when we remove cilia from the hippocampus, we had deficits in spatial memory, even the long-term memory. So it's interesting to see how cilia in different sides of the brain might play different functions. And I would like to thank members of my lab who participated in these studies, and my collaborator, Dr. Olivier Chivili, who is here, and Dr. Pierre Baldi. Thank you.
Once again, I think we have a few minutes for questions. And I see we have one already.
Yeah, question. Do these, the microtubules in the cilia also have tau protein? Are there any disorders that you've seen of the tau protein in these microtubules?
So my knowledge is that tau protein is mainly the microtubule that's cytoskeletal microtubule attached protein. But so far, I don't think there is evidence that tau might be linked to cilia that protrudes outside the cell.
Thank you.
That leads directly to a question I'd like to ask. Do the cilia that you're describing all extend outside of the cells? They do.
Yes.
And do they, now historically, among the early single-celled animals, I guess they were eukaryotes that had cilia, they were involved in motion.
Yes.
Is that still true of the cilia you're describing, that they have a mechanical operation?
So as I mentioned in my first slide, there are two types of cilia. So the motile cilia, which are 9+2, those are the ones that are used for moving. And we have them in the brain, but those exist around the ventricles to move the fluid, the cerebrospinal fluid. However, the cilia that I deleted or I am focusing on are the primary cilia, and those don't move. They bend, but don't move.
I'd like to save questions for later because I could ask several more. Any other questions? Well, thank you so much. And now we have a speaker who came to us from far away. This one came from Italy, Dr. Ruggiero. And he's a retired diagnostic radiologist from the University of Firenze in Italy. And his topic is the brain microbiota as it relates to the Orch OR theory of consciousness. I need to move the microphone.
Yes, I do. Can you hear me? Okay, thank you so much for this beautiful introduction. Actually, I moved to Arizona about 7 years ago after my retirement from the University of Florence. And let's see if I can open this presentation. Okay, so this is the title of my presentation today. Before I start, I wish to thank the organizers, in particular, Mrs. Bihar Montefiore, Professor Hameroff, and all the organizers and the technical staff of this wonderful conference. And also I wish to thank in advance all those who will have the patience to listen to this presentation. Some of the concepts, but not some of the newest results, have been published in this article about 1 year ago in the Journal of Neurology and Stroke that has the same title of my presentation, The Brain Microbiota and the Orch OR Theory of Consciousness. Now, I think that everybody attending this conference knows what the Orch OR theory is. So I am the least qualified person to talk about the theory in itself. What I've learned that the common denominator of the various evolutions and revisions of the theory is the concept that the consciousness arises inside brain neurons as a result of quantum computing occurring in the context of microtubules. And maybe we're learning also in the context of DNA or also in the context of cilia. Now, if quantum computing in cytoskeletal structures is the key, then in my opinion, there is no theoretical impediment at hypothesizing that cytoskeleton-like structures of microbes residing inside the human brain could give rise to forms of consciousness. So it could be a cytoskeleton-like structures like tubulin-like molecules of microbes, or could be cilia of microbes, and why not DNA of microbes? But are there microbes inside our brains? Yes, there are. Because far from being sterile, the human brain has a well-recognized microbiome. This was first observed in 2013 by Canadian researchers who were studying brains of people who had died of HIV/AIDS. And to their great surprise, they found that also healthy people, brains of healthy people, they had microbes, a very restricted but distinct bacterial population, which was composed of microbes that you commonly find in soil and water. And we continuously breathe, eat, and drink those microbes. So in our brains, we have the microbes that are in the environment. And how do they arrive to the brain? They arrive carried by cells of the immune system, mainly activated T lymphocytes and macrophages that work like Trojan horses, bringing these microbes from the environment, from the intestine, from all parts of our body inside our brains. What do they do, the microbes in our brain? Well, we don't know exactly, but since 2013, it was thought that their influence on brain function is nothing less than immense. So these are their words. And about 10 years ago, they postulated that the study of the brain microbiome or microbiota, the difference between individuals and the difference in different species might lead, as they write, to greater understanding of human neurobiology, including cognitive, motor, sensory, and behavioral functions, and I add, as well as consciousness. Now, about 3 years later, in 2016, it was observed that the brain microbiome changes with disease. So there is a dysbiosis in the brain microbiome, just like there is dysbiosis in almost all other human diseases, from the gut to the skin. Every time there is a disease, there is a concomitant dysbiosis. In this case, there was a reduced molecular diversity of bacterial RNA sequences in brains of people with multiple sclerosis. And as these authors wrote, these results resemble changes in microbial populations in other diseased organs and are reminiscent of a dysbiosis that can be resolved by increasing microbial diversity. So today we are familiar with the concept of microbiome medicine. That is, by modifying the microbiome or by rebalancing the microbiome that has been modified as a consequence of a chronic disease, you help with the disease itself. So why not also with neurological, neurodegenerative, neurodevelopmental diseases, since the brain microbiome is affected by the disease? But we can also think the other way around. Is the disease because of a dysbiosis of the brain microbiome? Well, you know, this has not been clarified yet, but I think that it is interesting, this comparison between dysbiosis in the brain and dysbiosis in all other organs affected by chronic conditions. This is more recent, 2018. These researchers from Birmingham, Alabama, they checked for the density of the bacteria, and they saw that the bacteria are not distributed equally in the brain, but some regions like the substantia nigra or the hippocampus or the prefrontal cortex, they have a larger number of bacteria. And also the bacteria are inside the cells, in particular glial cells like the astrocytes. Now, beside the consideration that the capacity of bacteria for influencing brain function is nothing less than immense, what could be their role in the context of neural connections and therefore of consciousness? So this was the goal of our experiments. And in our experiments, we were inspired by this very old observation that bacteria, they influence the social behavior of eukaryotic cells. So according to this old article, bacteria transform the closest living relatives of animals from single cells into colonies, from single to multicellular organisms. So we did some studies in the laboratory, and we used petri dishes with human neuronal cells, the SHSY5Y cell line, human neuroblastoma cell, and microglial cells. Now, as far as the bacteria were concerned, we decided to use a complete microbiome, not one strain of bacteria, not two strains of bacteria, but a complete microbiome. For those of you who are not familiar with this concept, a microbiome is composed by the microbiota, that is the microbes themselves, plus their theater of activity. So, for example, let's say you want to buy a probiotic, you go to your local grocery store and you buy 5-6 strains of bifidobacterium, lactobacillus, streptococcus. And those are the bacteria. You can also buy yeast, of course. But a microbiome is a complete entire ecosystem where you have the microbes plus their theater of activity, which includes protein, peptides, polysaccharides, but also phages, also plasmids, and also all the microbial metabolites that are produced by the microbes. So in essence, this is like a yogurt or a kefir, where you have the microbes plus all the molecules that they have produced, modified. And so it's a much more complete ecosystem, a complete ecosystem. And we have used an ecosystem that we have developed and we have characterized, we have published, in particular, as far as the composition in phages and plasmids are concerned. These are the two articles where we describe the complete characterization of this ecosystem. Now, these are the human neuronal cells on a petri dish. They do little. They emit some elongations that could resemble like axons or dendrites, but they don't do much. They just sit there and they replicate very slowly. Then we did something that is anathema for all cell biologists. That is, we threw a microbiome, that is, we threw bacteria plus all their molecules into the petri dish. This is something that is absolutely forbidden in the minds of everybody who does a cell culture. And to our great surprise, the cells, they did not die. Actually, in this petri dish, the little tiny dots that you may see, those are the bacteria. And as you can see, the human neurons, they have changed completely their morphology and their connections. because they have changed their shape and now they connect with each other, forming a series of circuits, long circuits, short circuits, so a series of connections. This is another field where you can see the tiny dots of the bacteria and the change in the shape and in the social behavior of the human neurons. Then the next step was try to start from somewhere, identifying which molecules could have been responsible for this phenomenon. Just to give you an idea, if you take a typical bovine kefir, that is a kefir made with bovine milk, you end up with at least 236 unique peptides. Unique peptides means they were not present in the bovine milk. The microbes, they made these peptides essentially out of casein. And each one of these peptides does something. Essentially, they promote health through a number of functions. Now, of course, we could not analyze 236 peptides at once. So we started from somewhere. And we started from a microbial metabolite that we knew very well for other reasons, because it is a powerful immune stimulator. And it is a vitamin D binding protein derived macrophage activating factor, also known as GC-MAF, macrophage activating factor. We were studying this protein, this peptide, in the context of chemotherapy, and in particular of chemotherapy with oxaliplatin that is known to cause neuronal damage. So we wanted to see whether this peptide, GC-MAF, was able to counteract the neuronal damage that is known as a side effect of oxaliplatin treatment. And by the way, it's interesting because this is a macrophage activating factor, and macrophages are instrumental in balancing the brain microbiome because they carry the microbes from the gut, from the environment into the brain. So these are, again, the control neuronal cells in culture. And again, they do what they usually do. They establish a few connections. And this is the way they look after treatment with GC-MAF. So here there are no microbes, but only one of these more than 200 peptides produced by the microbes. And so you can see again the change in shape and the increase in number of connections. And again, another field. Now, then we did something else. We co-incubated neuronal and glial cells. The neuronal cells are those pale pink here, and the glial cells are those darker. These are the controls, so they simply sit there, and this is what they do after they have been treated for 24 hours with GC-MAF. So again, they change shape, but most important, they change their social behavior, and now they form a very clear circuit. Now, when we do microscopy, both conventional electron microscopy, and also when we do radiology, we are biased by a conscious or unconscious desire of observing what fits with our hypothesis. We knew this very well. And so because of this, we developed an automated computerized analysis of cell morphology that measures elongations between neurons and also between neurons and glial cells that could resemble neurites and dendrites. As a matter of fact, such an analysis was originally developed by some engineers working with us in university for another purpose, to study the effects of ultrasound on human neurons and microglia. So this might be of interest to those who study the effect of ultrasound on consciousness. So this is the way the machine does. It's not artificial intelligence. I mean, it's an algorithm, not too sophisticated. I could understand it, but at least it is automated and objective. So it removes the conscious or unconscious bias of choosing the fields that were more interesting for us. So we could determine the increase in connections in the elongations with this automated machine. And these are the articles where all this has been published. Now, you may wonder, what has all this to do with the Orch OR theory? So what is the relationship? What could the brain microbiome add to the Orch OR theory? In my humble opinion, it could add a lot. For example, this is one of the latest articles that I read on Orch OR theory, how to falsify the theory or to prove it right. for example, using anesthetics. And also the previous speaker has used anesthetic gases as a mean to prove the involvement of microtubules. Well, this is a very good idea, but we have to consider that volatile anesthetics, they do affect bacterial functions such as motility, biofilm formation, and signal transduction. Therefore, in designing those experiments to prove or falsify Orch OR, the effects of volatile anesthetics on the brain microbiota should be also considered, so to prevent potential misinterpretation of the results. And I think it's particularly interesting to consider the effects on biofilm formation, because it is considered that biofilm formation is paradigmatic of the so-called microbial intelligence. This is an article published in 2014 where biofilm formation is taken as a paradigm demonstrating microbial intelligence. Now, the concept of microbial consciousness is a rather old one. And according to an article published in 2019 in Bioessays, microbial consciousness arose from cytoskeleton-like structures, and it emerged as an intrinsic feature of the first unicellular forms of life through the working of oscillating cytoskeletal structures and structurally flexible proteins. And I think that you recognize that it is very similar to the concept at the basis of Orch OR. Now, another consequence of considering the brain microbiome is that if the phenomena of quantum entanglement occur between the microtubules of adjacent neurons, they may occur also between the cytoskeletal structures of human neurons and those of the brain microbes, as well as between the cytoskeleton-like structures of the different microbes. And therefore, the human and the microbial consciousness are potentially entangled. Entanglement of human and microbial consciousness is all the more conceivable when considering that wireless communication, this comes from Professor Hameroff's papers, wireless communication based on the resonance of vibrations inside microtubules does not require neurotransmitters or synapses, because of course the microbes, they are not connected to the neurons with synapses. But since this is a wireless communication, it can occur between neurons and microbial cells, even though they are not anatomically connected by synapses. And if the concept of the brain microbiome and the ensuing concept of the rhizome, and here I'm talking about a philosophical concept of rhizome as originally proposed by the French philosophers Gilles Deleuze and Félix Guattari. So if this concept could be integrated in Orch OR, the explanatory and predictive powers of the theory would be vastly increased. If you want to have fun, you can read this little essay that I wrote on the human microbiota, the immune system, and immortality. Why immortality? For example, the issue of permanence of consciousness independently of neurons would be further supported by the consideration that the microbiome survives our human body. When we die, the microbes, they don't, actually they thrive. So if the consciousness has become entangled, this could explain a number of things. Because if the Orch OR phenomena occurring inside the microtubules of human neurons are indeed entangled and shared with those of the microbiome, we should remember that the microbiome is an organ that has 10 times more cells than our human cells. But when we talk about genes, we are made of 22,000 genes. The human microbiome is made of 232 million genes, or maybe a number of genes that is higher than the number of atoms in the universe, and this comes from Harvard Medical School. So when the information is entangled and shared with such a big organ, then I think the survival of consciousness might become a topic pertaining to microbiology as well as to philosophy. In conclusion, and I hope that I'm on time, just as it would be unrealistic today in 2022 to discuss the development and function of all organs, from the gut to the immune system, without considering the essential role of the microbiome, here I propose that also the discussion on human consciousness, and in particular on Orch OR, should take into account the role of the brain microbiome. And thank you so much for your attention.
Once again, I think we have time for questions.
It's fascinating. I've been a physician for 40 years. I've never heard this about microbiome in the brain. But I do know that in the gut and in the mouth, at least 50% of the bacteria belong to the archaea class of bacteria, which is a phyla of which no member belong to phyla that no member has ever been cultured. Could there be these archaea bacteria also that are having a role? And would you have any idea about that?
Well, the few studies that I've done, because you're not the only one who has never heard about the brain microbiome. Very few people, I don't know why, study this topic. Well, one reason is that you can easily study the bacteria in the gut, in the stools, I mean, and you can measure and you could do studies. Much more difficult is to study the microbes in the brain. They're mostly alpha proteobacteria. And this is interesting because they do contain pigments. So today we heard that pigments are molecules very much involved in these quantum phenomena that might be at the basis of consciousness. Archaea have not yet been identified. I cannot rule out that they do exist, they are there, but as yet they have not been identified.
Thank you.
May I chime in with a question? It's my understanding that microbes typically evolve much more rapidly than large organisms like us. Does this mean that the evolution of microbes in the brain could be occurring and could be modifying the operations of our brain?
Yes, this is quite possible. As a matter of fact, in multiple sclerosis, for example, the biodiversity of the brain microbiome is lower, as it happens in a number of diseases of the gut. For example, when you have a chronic bowel inflammation, you have a much less diverse microbiome. That's why probiotics, they're used, because they reconstitute the microbiome, they reconstitute the diversity, the richness of the microbiome. So it is quite possible that the role of the microbiome in the brain is exactly as it is in the gut, in the immune system. And since microbes, as you correctly said, they divide much more rapidly than human cells. Our neurons, they divide, but only a little bit and quite slowly. Microbes, they divide much faster and they adapt much faster. It is quite possible that their role is much, much greater than we have even suspected.
Thank you so much. Thank you. According to the official schedule that I received, the next speaker is Mr. Davis, but I do not have any word from him. I'm going to defer to our tech. Do you have any indication, Matt, that Mr. Davis is? I don't find a video from him. And I didn't see any indication that he's joined us on the Zoom.
Oh no, okay, he's on Zoom. Yeah, I don't see it. Let's move on to another speaker, and I'll double check that nothing got uploaded.
If Mr. Grinde is ready, he would be next. And I'll just quickly introduce him. Mr. Grinde comes to us. I think you are coming from Europe, right?
Yes, I am.
Okay. From the Norwegian Institute of Public Health. His topic is another one related to evolution. Did consciousness first evolve in the amniotes?
Okay, thank you. I'm a happiness biologist, which means I try to understand why we have the capacity to feel good or feel bad, and what goes on in the brain when we do that. And I can work on that because my institute has considerable engagement in trying to help out the mental health problem. And we believe that by understanding happiness, we can perhaps alleviate mental health issues. And as feelings are a core part of experiences, which are what makes up consciousness, I believe that to understand feelings, it's also useful to understand consciousness. And these two are actually related in evolutionary terms, I believe. So the topic today is why and when did evolution add consciousness to animals? And as to the when, I believe the answer is that this started some 300 million years ago in amniotes, or early amniotes, and amniotes are the shared ancestors of reptiles, birds, and mammals. And I'll use this opportunity to offer the evidence that is behind this idea. Well, I know there are lots of different opinions as to what evolution is about at this conference. And so some people might think that plants like this rose have consciousness, and I would say, I'm sorry, I do not think so. In order to have experiences, you need a nervous system. So how about the nematode then, this tiny little worm? Can it have consciousness? And again, I said, sorry, but you need a large and advanced nervous system, and the nematodes only have a few hundred neurons in their ganglions. On the other hand, we know that humans have consciousness, and that means that this feature entered the lineage at some point between the split between nematodes, so simple organisms, and the vertebrates. So consciousness is a feature of advanced nervous systems. And it is useful to take a short look at what nervous systems are about. And their purpose is to conduct behavior, and behavior generally just means movement. And all nervous systems are based on the same kind of template. You have a sensory arm that registers things in environment or in internal body. And then there is a processing unit that uses this information to decide what to do. And then there is the effector unit that activates the muscles and cause movement. And of these three components, the processing unit or brain or ganglia, depending on the organism, is the most complex and the most interesting part. And but even the very simple, easy or small processing units, such as those of the nematode you saw before, they can learn, they can to some extent have attention, and they make decisions in the meaning that they do move in different directions depending on sensory stimuli. So a core question then is why move? And I mean, plants don't need any nervous systems because they don't need to move. As long as sunshine comes from above, they can just stay put at one spot. But movement for animals are about two core issues. You either move away from what is bad for your genes, or you move toward what is good for your genes. And this is the same little nematode swimming here toward the ice cream. And in these processing units or ganglia or brains, evolution added a lot of different types of algorithms for making decisions. The simple forms are just reflexes or instincts, and also learned actions like this nematode can have. And then feelings and consciousness, I believe, is best understood as an advanced way of making decisions, an advanced algorithm that came in certain organisms because it helped them to make the more optimal behavior. And while looking at the different types of animals present today on Earth, just 3 of the some 44 different phyla or groups of animals have advanced nervous systems. That's the arthropods, such as the ant and the mollusks, here with an octopus, and then of course the vertebrates. So then one question is, could consciousness as a strategy for behavior be present in all these three groups of animals? And my answer is that is very unlikely, because going back, their shared common ancestors dates back to almost the first nervous systems, close to 600 million years ago, at a time when the system presumably was at least as simple as that of the nematode. So in order to have this specific type of strategy in all these phyla would require a very strange and unexpected form of evolution towards similar structures. And that, of course, you could have what is called convergent evolution for this purpose to establish consciousness and feelings. But unless the conscious strategy is a very brilliant one or very functional, that seems unlikely. And I might offend somebody here, but consciousness is not really that great at all. And I can kind of document that by looking at the evolution of organisms. For one, the most successful type of animals are the arthropods, such as this little shrimp and the ant. And as measured in biomass or measured in variety of species. And actually, if you look at biological success, which is typically measured in biomass, we find that the most successful animal on Earth is this little shrimp here. Its biomass, the combined biomass of all these shrimps is about twice that of all humans. And furthermore, if you look at our evolutionary tree on the left here, we see that going back just about 100,000 years, there were some 7, 8 different species or subspecies of humans, and all but one of these are now extinct, and they were equally, presumably had the equal intelligence as we have, and in fact, the Neanderthals had larger brains than we do. And the same goes for our close relatives of animals, the chimpanzee and the gorillas. They're not doing very great, biologically speaking, and some of these species are actually on the verge of extinction. So I mean, biologically speaking, consciousness is not that great, and actually even in our lineage, the success is very recent and has nothing really to do with, or it's not the direct consequence of a genetic makeup. It's a cultural thing. That came like long after we developed our genetic constitution. So yes, consciousness do have advantages. It can give you the flexible and adaptable behavior strategies, and also it can cause a very complex behavior, like in humans with a complex language and thoughts and deliberations. But there are also very obvious disadvantages of introducing this kind of strategy for making decisions. It's very slow compared to non-conscious strategies, extremely slow. It's very energy demanding, which means it's restricted to certain types of decision making. Most of what goes on in even the human brain is non-conscious. And it can only focus at one thing at a time. We may shift between different tasks, but unlike the non-conscious nervous system, you can only take on one thing at a time, while the unconscious system can deal with lots of issues simultaneously. So it is for decisions, but not for execution of these decisions. Even just talking like I do now requires the use of the movement of some 300 muscles per second, and consciousness could never manage to organize that. I can only direct the brain to what I want to say, and then unconscious, non-conscious processes takes over and actually move the muscles. And it's vulnerable to whims. We may, for example, decide we don't want any children, which is good for society, but very bad for the genes. So although convergent evolution do take place, like, for example, in the wings of insects and birds, in this case, in the case of wings, it is a very obvious functional thing. and wings need to have about the same shape in order to enable flight for an animal. While it seems unlikely that the three phyla with advanced nervous systems should end up with the same type of strategies for behavioral control. For one, as I mentioned, because consciousness is not really worth it. It's not that a good strategy. And so to understand kind of under consciousness, I think it's useful to try to consider what sort of strategies could you program in a computer, because the evolution presumably can make about the same type of strategies in an unconscious, non-conscious brain as you can make in a computer. And you can make very advanced behavior without consciousness. But there's one thing you cannot do, and that is causing experience. You can program a computer presumably to simulate having experiences or feelings, but it won't have any experiences or feelings. At least in my mind, it won't or cannot have. So there must be a reason why this particular type of strategy for making decisions did evolve. And I think the key to understand why consciousness started its evolutionary history is because it is connected with the need to feel, and feelings are really just a strategy for making decisions that are built on punishment and rewards. And brain punishments, bad feelings, is about trying to make you avoid what is bad for the genes, while the positive feelings are about trying to make you do what is good for the genes, which is why feelings have two kind of shades or two main types. So what feelings is about as a strategy is to make decisions based on a common currency. The brain tries to weigh the positive sides against the negative sides, and then tries to go for the maximum positive outcome in its decisions. And consciousness, I believe, came as a consequence of evolution starting on this strategy, because feelings does require the capacity to feel, and it's kind of, in my mind, the only type of brain processes that could not be taking place in either a computer or in a non-conscious system, which makes me believe that this is what actually caused the start of our conscious experiences, that we needed the capacity to feel in order to use feelings as a way of making the right decisions. So I kind of tried to say that consciousness is only present in vertebrates, and then the next question is which vertebrates can have consciousness. And it's kind of, we can recognize signs of having feelings in reptiles and birds and mammals, but not the same in amphibians. For one thing, we can see that these reptiles that do react physiologically to handling in ways that are expected from an animal who do feel something. They have rapid heartbeat or increased temperature if you pick them up, while amphibians do not. They have a apparent capacity to feel pain. I mean, any organism any animal will look like it feels pains because it has aversion reactions, but there's science that the amniotes actually can feel the pain. And they have behavior indicative of emotional evaluation. We can also see on their use of specific neurotransmitters and the structures in the brain that are engaged with these neurotransmitters, that they are similar to what we do when we experience feelings. We see the same patterns in these animals. And we see the brain structures that are kind of involved in human feelings in these animals. And we also find that reptiles and birds and mammals certainly birds and mammals, but not amphibians, have REM sleep, which is a kind of conscious, semi-conscious state in mammalian sleep, which is then again a sign that consciousness is likely to be in some form present in these other animals as well. So that kind of means that feelings started to evolve in the amniotes, and the reptiles and mammals split off a bit more than 300 million years ago. So I would restrict consciousness to these groups of animals. And there's actually another reason why you'd expect the amniotes, the early reptiles, to evolve new or novel strategies for behavior. For one, they started out with quite complex nervous system compared to other animals that are present on out of the water. Like insects cannot grow large and they have very small nervous systems. And also the reptiles developed lungs, which means they can gather a lot more oxygen from the air than any aquatic animal can take from water, which again translates into having much more energy to spend on this expensive new organ, the brain. So we know that there was a considerable expansion in nervous system upon the adaptation to living on land compared to being in the water. And also that would be because there's a rapidly changing habitat. In the living in the sea is pretty much the same year round, the same environment around you all the time. While when you move onshore, then it's a much more variable environment, and that requires to have more adaptable behavior. And of course, the reptiles, certainly compared to insects, are very large animals. And with the short or few, they don't make much progeny. So they need kind of to adapt individually rather than to have evolution change their constitution. So these kind of factors that are pointing towards the idea that consciousness started in these animals. And of course, we humans, we consider our consciousness have lots of features that the early reptiles certainly did not have. So we feel consciousness to be a lot more than just behavioral strategies for making decisions. We can have a very advanced film of life, and we have self-awareness, that again gives us a fairly good dose of free will. And we have deep insights that can help us to make even more advanced decisions. But the basic thing, the basic form of consciousness started presumably in the early reptiles. So just briefly at the end, we think consciousness is such a fantastic thing about the brain, but I think it's a minor thing in the brain, even assuming that all mammals have consciousness. It means that even the shrew with a brain of just 60 milligram would be able to have consciousness, which seems likely, while the octopus with a brain of 2 grams does not need to have consciousness to make complex behavior, because even it's very complex behavior you find in octopuses, they can still be thought, there's no reason why they could not be programmed into the nervous system of these animals. And even humans have a stomach brain in the form that the neurology associated with the guts have a total weight of about 1 gram, but that's not conscious either. So size is not important. It's just that it's more the complexity of connections between the neurons that seems to be what is necessary to have consciousness. So I think rather than consciousness being such the most complex function we find in the human brains, I think other functions we have are more difficult to set up, require more neurology. For example, muscle coordination, which is cared for to a large extent by the cerebellum, which again contains about 80% of the neurons of the human brain. And then again, things like being able to decipher sound or sight also are very complex matters. That's fine. This is my last slide, just to remember that consciousness, it is after all you have, but it's really only ripples on the ocean of brain activity. Thank you.
I think we have time for just a couple of short questions.
Well, I'm way out of my expertise asking this question, but I thought there was some evidence that fish do feel pain to do with giving them anesthetics under, or they tend to go to a situation where an anesthetic, a local anesthetic is in the water, and that enables when there's a painful stimulus in some other water. And also on the question of rapidly changing environment, I'm not quite clear how being in the sea with lots of other types of fish and plants and things is less rapidly changing than being on land. But I'm not trying to challenge what you're saying, which I really liked, but just a matter of information.
There's quite a few reports saying that both lower vertebrates and invertebrates do have feelings, because we do see them behave somewhat like they had feelings. And what is problematic with that research is that they often you can use substances like biodisciplines, like substances that are anxiolytic in humans, and actually they will also affect behavior in these lower animals. But the point is that that is because the basic neurotransmitters are with us from the very earliest nervous systems, and they do have kind of conserved function. So avoidance behavior is likely to be under the control of similar neurotransmitters as fear and anxiety is in mammals. And then, so it looks like you can treat, so this medicine that affect these neurotransmitters will tend to be, to also affect similar behavior in these animals. But that doesn't necessarily mean that you feel the stimuli. And yes, I mean, well, the ocean has been the same, or it's been slowly evolving for millions of years. On land, I believe anyway, it was more rapid changes going on compared to what the typical organism in the ocean were used to, partly because land was colonized at this time, and it was a kind of new habitat with very much diversity of animals taking place and much diversity in species. And of course, water is not always accessible on land, but it's always accessible in the ocean and so on. And temperature is about the same all the time. Okay. Well, thank you.
And I would like to maybe ask some questions after the session, Mr. Grinde. At this point, we're ready to begin with two participants who are joining us through Zoom. And I'm just going to make a slight explanation of, I don't think we have any other live in-person participants. I learned from Matt that we have a third video besides those of the two participants. And I suggest, and that came from Mr. Davis, I suggest for those who are interested, I suggest we play that at the end of the session, which means that our next participant is Alexander Egoyan from the Georgian State University of Physical Education and Sport. And the title is Concepts of Elastic Membrane and Elastic Quasi-Membrane as Complementary Tools for Understanding Consciousness and Brain Function. Are you able to bring up?
Yes. Hi, my name is Alexander Egoyan. I am Associate Professor at the Department of Biomechanics and Computer Technologies at the Georgian State University of Physical Education and Sports. First of all, I would like to thank the organizers of this excellent conference for inviting me here. The science of consciousness is different from other sciences because it unifies natural sciences such as physics and biology with psychology and philosophy. The science of consciousness involves two approaches, the traditional physical approach and the phenomenological approach, which allows us to study our perceptions, feelings, and emotions directly from our own phenomenal experiences, just like Gestalt psychologists do. The question is how to unify these two approaches. It is clear that instead of using one theory, we should use two complementary theories. Unfortunately, we have very well developed physical theories, while there is not a physical theory explaining holistic, macroscopic, and multidimensional properties of qualia. It is obvious that the best way to create such a theory is to introduce new holistic, macroscopic, indivisible objects and try to explain qualia using these objects. In my previous works, I have introduced the concept of elastic membrane. Elastic membranes may be described mathematically as there is a new elastic geometry behind them. In this geometry, each elastic membrane may be described by a surface which has two metrics, the internal and external metrics. This means that the membrane may change its form in the higher dimensional space without changing the internal metrics. This theory is multidimensional, just like our consciousness is. This slide shows how a two-dimensional elastic membrane changes its form after elastic deformation. This slide shows deformations of a fragment of a two-dimensional elastic membrane after stretching, local squeezing, and curving.
Can you hear me?
Yes. Yes.
Could you share your screen so that we can see?
Oh, excuse me. Excuse me. Excuse me. So, excuse me. My name is Alexander Egoyan. I'm associate professor at the Department of Biomechanics and Computer Technologies at the Georgia State University of Physical Education and Sport. First of all, I would like to thank the organizers of this conference for inviting me here. The science of consciousness is different from other sciences because it unifies natural sciences such as physics and biology with psychology and philosophy. The science of consciousness involves two approaches, the traditional physical approach and the phenomenological approach, which allows us to study our perceptions, feelings, and emotions directly from our own phenomenal experiences, just like Gestalt psychologists do. The question is how to unify these two approaches. It is clear that instead of using one theory, we should use two complementary theories. Unfortunately, we have very well developed physical theories, while there is not a physical theory explaining holistic, macroscopic, and multidimensional properties of qualia. It is obvious that the best way to create such a theory is to introduce new holistic, macroscopic, indivisible objects and try to explain qualia using these objects. In my previous works, I have introduced the concept of elastic membrane. Elastic membranes may be described mathematically. There is a new elastic geometry behind them. In this geometry, each elastic membrane may be described by a surface which has two metrics, the internal and external metrics. This means that the membrane may change its form in the higher dimensional space without changing the internal metric. The theory is multidimensional, just like our consciousness is. This slide shows how a two-dimensional elastic membrane changes its form after elastic deformation. This slide shows deformations of a fragment of a two-dimensional elastic membrane after stretching, local squeezing, and curving. The theory is complementary to the theory of general relativity, according to which space and time are not curved. Gravity is a consequence of the changes in the surface density of the elastic membrane caused by elementary particles embedded in it. It is well known that the theory of general relativity is multidimensional. The number of space dimensions is not fixed in it, but it does not explain how space times having different numbers of space dimensions can be embedded one into another. The elastic membrane theory solves this problem. From the outside, elastic membrane is a material object, while for the internal observer, it is a space-time we have used to. Elastic membranes may be two-sided or one-sided, having material properties on both sides or only on a single side. They have no thickness. They are similar to the brains from string theory. This picture shows how gravity may be encoded into the surface density of elastic membrane. Even our universe can be considered as a flat, infinite membrane moving along the time axis. This model requires the preferred reference frame where two-dimensional elastic membranes are embedded into the three-dimensional elastic membrane of the universe. Two-dimensional elastic membranes with closed topology may be interpreted as perceiving entities embedded into human brain. Only one elastic membrane will correspond to the selected organism. According to the elastic membrane theory, we are two-dimensional elastic membranes where perceptions are caused by vibrations of dense enough regions of the membrane. Frequencies of the oscillations are supposed to be responsible for the quality of different perceptions, colors, sounds, etc., while amplitudes for the feeling of being three-dimensional. Each elastic membrane may be considered from 3 points of view. First, from the point of view of the external observer, it is a surface with closed topology. Second, from the point of view of the internal observer, it is a space-time continuum we are used to. And third, from the first person's point of view, being an elastic membrane. A two-dimensional elastic membrane can feel only deformations which contain squeezing. It cannot perceive its own form, shape. This picture shows how elastic membrane, one-dimensional elastic membrane, changes its position. It has two parts. One part is marked by green markers, and the second part is marked by red markers. When one part is moving, another part is in rest. Elastic membranes move with the brain in the preferred reference frame, but their motion is different from the motion of other physical objects. Each elastic membrane may be considered being in rest, just instantaneously changing stationary positions from time to time. In other reference frames, elastic membranes become quasi-membranes, which are not real objects and may be associated with Platonic values embedded into the space-time geometry. Due to their elasticity, each elastic membrane will contain motionless and moving parts. In the case of elastic membranes, the accent is made on the motionless parts, while in the case of elastic quasi-membranes, on the moving parts. This means that when observed in the preferred reference frame, each elastic membrane may be considered being in rest, just instantaneously changing stationary positions from time to time, while each elastic quasi-membrane is moving all the time, changing its moving parts and having its own space-time geometry. These two concepts give rise to two complementary theories. The first one is phenomenological, suitable for a qualitative explanation of perception, while the second one is physical, capable of explaining real physical phenomena in the brain. According to the first theory, the elastic membrane squeezes around thalamic nucleus from where it propagates through the neural network to the regions of the cortex responsible for perception and tries to occupy most stable positions around the brain microtubulus. This slide shows how elastic membrane, shown by red, occupies most favorable positions in the brain. It can propagate through the axons of single neurons, as it is shown on the right, or it can surround groups of neurons, as it is shown on the left. This picture shows how elastic membrane propagates from one neuron to another neuron. It is supposed that our perceptions are encoded into the vibrations of the elastic membrane. Only squeezed regions of the membrane produce perceptions because the membrane should be dense enough to generate perceptions. Squeezing of a region of the membrane happens at the expense of stretching of the surrounding regions. From the squeezed regions, perceptions are projected on the stretched regions. This model explains how vibrations of different neurons of sensory perceptual cortex give rise to qualia. This picture shows how the elastic membrane propagates through a neuron. You can see squeezed regions here, which are responsible for perception. Elastic membranes squeeze around brain microtubulus because they are the most stable symmetrical structures in the brain neurons. The elastic membrane propagates in the gravitational field modulated by microtubulus. It is trying to find the most balanced position in which elastic, gravitational, and electromagnetic interactions are in stable equilibrium. In this position, the membrane will produce collective vibrations around brain microtubulus responsible for qualia. This picture shows how elastic membrane squeezes around brain microtubulus. Elastic membranes are very similar to quantum computers. They are whole things. All regions of the membrane stay interconnected during elastic transformations of the elastic membrane. This means that the information exchange through the elastic membrane happens instantaneously. Its parts stretch and squeeze in a coherent way. This is why quantum computers seems to be the best tool for processing the signals coming from the elastic membrane. Regions of the elastic membrane squeeze around brain microtubulus in a coherent way, trying to optimize the process of membrane deformation. According to the second theory, the elastic quasi-membrane separates two regions of the brain. The region surrounded by the elastic quasi-membrane.
Mr. Egoyan?
Yes?
We're coming
Can you hear me?
We're coming close to the end of your time.
Few minutes, 5 minutes. I need 5 minutes. And the region outside of the elastic quasi-membranes are not real objects, and their role in brain processes is supposed to be similar to the role of quasi-particles, phonons, magnons, and excitons in the solid state physics. The notion of elastic quasi-membrane is derived from the notion of elastic membrane. Therefore, the interactions between the two regions should happen in a holistic way and have an elastic character. Furthermore, these interactions will happen in the brain microtubulus. The elastic quasi-membrane passes through and will be modulated by Platonic values embedded into the space-time geometry. This means that interactions between the two regions inside different neurons will occur simultaneously and will be interconnected. It is obvious that quantum computations are needed to support such holistic interactions between the two regions. According to the theory, our perceptions are caused by macroscopic brain phenomena, which involve a large number of neurons working in a coherent way. Holistic interactions in microtubulus of different neurons require quantum computations. Elastic membrane concept supports all axioms of IIT. Intrinsic existence. Elastic membrane exists. It is as a physical object. Consciousness is structured. Elastic deformations are also structured. Consciousness is specific. Elastic deformations also are specific. Consciousness is unified. Elastic membrane is indivisible and therefore it is unified. Consciousness is definite. Elastic membrane is a physical object and therefore it has definite physical parameters. Elastic quasi-membrane concept is in agreement with Orch OR theory. It supports the exclusive role of brain microtubulus in formation of phenomenal experiences. It supports quantum computations. It admits the importance of gravitational curvature in the brain functioning and incorporates so-called Platonic values. It supports the wave function collapse theory of consciousness because it provides more stability for the system. It supports the idea that each consciousness state is bound to its own space-time geometry. The two theories are complementary and may be combined to find connections between phenomenological and physical models of brain processes. By using the first theory, we can try to find similarities between our perceptions and geometrical properties of elastic membranes. For example, we can use squeezing for interpretation of attention, vibrations for interpretation of different senses, stable collective vibrations for interpretation of pleasant perceptions, unstable collective vibrations for interpretation of unpleasant perceptions. From the first person's point of view, we can discover some new features of our perception, and then we can try to create elastic membrane models for them. Or by using the second theory, we can find elastic geometrical forms inside the brain and then try to connect them with our perceptions. Interestingly, the form of brain's convolutions is similar to the form of a curved elastic membrane. This method involves the detailed analysis of physics and geometry of the brain areas connected with the phenomenon we are interested in. The two methods are complementary. It is possible to check geometrical models developed using one method by means of another method. Pairs of complementary notions between the two theories. The elastic membrane theory is holographic, idealistic, multidimensional, elastic, deterministic, holistic, intuitive, and phenomenological. The elastic quasi-membrane theory is realistic, materialistic, three-dimensional, rigid, non-deterministic, reductionist, rational, and experimental.
I'm sorry,
Conclusion. In my talk, I have presented a powerful method for understanding brain processes and consciousness based on the concept of elastic membrane. The method is in a good agreement with mainstream theories of consciousness such as IIT and Orch OR. Moreover, it has multidimensional theories of geometry and physics behind it, which complement existing geometrical and physical theories, namely Riemannian geometry and general relativity. This gives the additional grounds in favor of my theory. Thank you for your attention. If needed, you can connect me on this email. Thank you.
me? Thank you, Mr.
Yes, 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 yes.
Unfortunately, I don't think we have time for questions until after the session, because we ran over on that one. Our last live person is Felice Todd of Pierce College, whose title is A Photochemical Signal Transduction Model of Magnetoreception in Cryptochrome.
Hello, thank you for having me. Are you able to play the 10-minute video?
You would prefer to have the video played and then take questions and answers?
Yes, please.
Can you do that, Matt?
We'll be discussing the sixth sense, a photochemical signal transduction model of magnetoreception in cryptochrome. In 2019, researchers at Caltech made a discovery that had long been suspected, but never conclusively detected despite prior attempts. They found that in addition to our 5 commonly known senses of touch, sight, smell, hearing, and taste, that we humans also have a sixth sense, known as magnetoreception. Magnetoreception is like a biological compass. It's the ability that many organisms have to detect and orient to magnetic fields. This sense is so sensitive that it can even detect relatively weak magnetic fields, like the Earth's geomagnetic field. Now, in the case of modern humans, it's unclear if this sense can be consciously used. The Caltech research only found that humans were subliminally aware of directional changes in an Earth-strength magnetic field. This was detected as alpha brainwave suppression, which is an indicator of sensory awareness. However, many animal species regularly use their ability to orient to the Earth's magnetic field for everyday survival. So how does this sixth sense work? Well, the precise mechanism underlying magnetoreception is still an active area of research. However, most evidence supports 2 possible methods, one classical, the other quantum-based, which may actually work together. The classical mechanism is attributed to magnetite crystals found in the brains of species like humans that exhibit magnetoreception. These crystals act like a physical compass. The quantum mechanism is a chemical compass where light activates a chemical reaction that is sensitive to directional changes in an external magnetic field. An analogy for how these 2 mechanisms might work together could be drawn from how we sense the sun. If you walk outside with your eyes closed, you can still sense the sun's presence as it warms your skin. But if you want to know exactly where the sun is oriented in the sky, say to determine the time of day, then you have to use your sight. Your eyes have to open to receive the sun's electromagnetic radiation. Similarly, a sight-based chemical compass is suspected to underlie magnetic field orientation. And it's this quantum-based model of magnetoreception that was the focus of my research as a doctoral candidate at the University of California, Irvine, under the direction of Professor Torsten Ritz and in collaboration with postdoctoral fellow Maria Procopio. So this is the work that I'll be discussing today. So why is a light-sensitive chemical reaction believed to be involved in the ability to sense the orientation of a magnetic field? Well, behavioral experiments in migratory birds suggest that their ability to properly orient to the geomagnetic field is light-dependent. Specifically, they can correctly navigate in a range from ultraviolet, blue, and green light, but not in yellow and red light. So let's examine how a chemical compass is proposed to function. Sunlight enters the eye and is absorbed by photoreceptors that are fixed within the cone cells of the retina. The cryptochrome photoreceptor has been suggested as a possible site for a chemical compass because it absorbs the UV, blue, and green wavelengths of incoming light, but not the yellow and red. The blue-green light sensor in cryptochrome is a flavin molecule that changes its shape from a non-binding state in the dark to a state that can bind with signaling molecules in light. Once in the signaling state, flavin activates the bound signaling molecule, a ligand, which then binds to a neural receptor and thereby transmits sensory information to the brain. Most crucially, the time flavin spends in this light-activated signaling state depends on its orientation within a magnetic field. So changes in magnetic field orientation may be signaled to the brain via fluctuations in the signaling state concentrations. The rates at which flavin cycles through each state is governed by both light-dependent rates and magnetically sensitive rates. This magnetic sensitivity arises when flavin, in its fully reduced state, donates an electron to another molecule like oxygen or tryptophan, thereby forming a radical pair. The lone electron spin of each molecule in the radical pair is correlated with the other in a quantum superposition of singlet and triplet states. These quantum states correspond to the relative orientation of the spins to one another, as depicted here. Let's examine more closely the case of a flavin superoxide radical pair. If both spins were only exposed to the external geomagnetic field, then they would precess about this field vector at the same rate, remaining in phase and antiparallel in the singlet state. However, unlike the superoxide molecule, the flavin experiences a local magnetic field produced by its own nuclear spin environment. This causes the flavin spin to precess differently around a resultant magnetic field vector, giving rise to spin orientations of quantum triplet states. As the eye moves within the geomagnetic field, so too does the flavin nuclear spin, which is fixed within the cell membrane of the retina. So when we calculate over time, the probability that the radical pair wave function will collapse into a singlet state, it is dependent on the relative angular orientation with the direction of the geomagnetic field. My research investigated whether a theoretical model based on flavin's known formation of radical pairs with oxygen and tryptophan could explain the light-dependent orientation of migratory birds that has been observed in behavioral experiments. I did this by modeling a photocycle in which flavin passes through these states, with the gray circular arrows highlighting magnetically sensitive singlet-triplet quantum superpositions for each radical pair. A signal-to-noise ratio, which indicates the sensitivity of this chemical compass model, is found by solving the first-order kinematics of the photocycle to steady state, with and without a change in magnetic field orientation. The findings are shown on this graph of all bird orientation experiments under various monochromatic light conditions published to date. The gray shaded region indicates the wavelengths and intensities at which the model predicts a chemical compass would function, which does correspond to the experimental observations in the ultraviolet, blue, and short green wavelengths. This chemical compass model may also have greater sensitivity to longer green wavelengths if flavin absorbs this light range more intensely than is currently indicated by in vitro measurements of its absorption spectrum, or if flavin forms additional magnetically sensitive radical pairs with other molecules. So what are the possible implications of magnetoreception for the furthering of our understanding of the science of consciousness? Well, firstly, a chemical compass may provide yet another example of how biological systems have evolved to sustain quantum superpositions on timescales significant enough to impact physiological processes. And secondly, the finding that humans unknowingly possess awareness of sensory information that other mammals actively use supports the importance of considering all evidence of human sensory experience, including that of non-local perception, as we seek to develop a complete model for a science of consciousness. In closing, I'd like to thank my collaborators, Dr. Ritz and Dr. Procopio, as well as to offer a special thanks to Stuart Hammerhoff, who first advised me to pursue research in quantum biology when I was a new graduate student attending my first Science of Consciousness conference.
Hello, everyone.
Thank you for listening, and I'm happy to answer any questions.
Thank you very much, Dr. Todd. I'm not sure what you can see. Can you see now, or do you?
I can share my screen if need be.
My main question is whether you'll be.
If there are questions, I can refer to any graphs if there are any questions.
The key question is, if someone is standing at this microphone, will you be able to understand their questions?
I can hear you fine.
Okay.
Hi. I was wondering, in the situation that's created with the sensing, is there a spinless state created? Like a Cooper pair type?
A spinless state. So if we look at the photocycle, the magnetic sensitivity arises when a radical pair is created. So here you have these two molecules, the flavin and tryptophan. That's sort of the ground state. And the photocycle is activated when blue-green light enters the eye and hits the cryptochrome and specifically hits this molecule, where the flavin then donates an electron to the tryptophan and moves it along to create this radical pair state. And then the lifetime of the radical pair, this is sort of the time scale at which that radical pair is alive and is cycling through these various quantum states. So I'm not sure if that answers the question. The lone spin is created when you go from the molecule to the radical pair. The radical pair is what has that lone extra spin that is then processing about external and internal magnetic field vectors.
Thank you.
Hi, my name is Mike.
Hi.
I liked your talk.
Thank you.
And so I've seen similar themes about the electron donation attributing to different phenomena like the ones you're discussing. And I was wondering if you had any experience in terms of discussing the purposes for inhibiting these electrons from being donated on like the larger scale types of cumulative behavior. The other day I saw something in a talk and I was going to ask a question about why he chose to inhibit electrons in this, in whatever his process was. And I was wondering if you ever thought about if thoughts about that ever came to mind for you during your research.
Not specifically, only because I was creating a theoretical model based on the experimental observations that we already have. So when it comes to the radical pairs that I investigated, we already have in vitro experiments showing magnetic sensitivity for these specific radical pairs, the flavin tryptophan and the flavin superoxide radical pairs. So I sort of took it from the peer-reviewed literature that magnetic sensitivity had already been established when those radical pairs were created with those lone electrons. In terms of your initial question of not creating lone electrons, not creating radical pairs, then you just simply wouldn't have the magnetic sensitivity. It really isn't when the flavin is in its ground state that you see that type of magnetic sensitivity. But again, just to distinguish between the experimental questions that have already sort of been answered, I would say that that's the case, which is sort of distinct from the theoretical model that I was developing, which says, okay, if you have this sensitivity with these radical pairs, given the photocycle, it's not clear if cryptochrome could form additional radical pairs. It's not clear if both radical pairs that I modeled are what is giving rise to magnetic sensitivity. But when I looked at those two radical pairs and that plausible photocycle based on the experimental evidence that we have, it does allow you to explain the behavioral experiments of light sensitivity in the birds, where the birds can navigate at low intensity blue-green light, and ultraviolet light, but they cannot navigate at high intensity light or in the yellow and red range. And you can show that just based on the quantum spin dynamics of that photocycle, if that answers your question.
Really cool. Thank you.
Yeah.
I think we have a couple more questions. Go ahead.
Thank you very much. A rather different sort of question. I think you said in humans, the magnetic sensitivity, is it always unconscious, or are there some people who claim to feel it?
So there are some earlier studies in the 1970s. I think the name is Baker. Those were the first experiments in humans where the humans actually were conscious of the directional change. Basically, he took some students and blindfolded them and drove them around in different convoluted paths, and then asked them to sort of reorient themselves. And in his experiments, he showed that people were able to correctly indicate a certain direction in those experiments. However, a subsequent experiment was unable to reproduce those results. So what is interesting about what the Caltech group did is they took out, they did not use a behavioral experiment per se. They actually looked at EEG brain scans, okay? And so normally when someone is in just a calm, resting state, maybe with your eyes closed, not deep meditation, but you're just resting, you're not particularly thinking, there's going to be a strong peak in your brainwave activity in the alpha region on a normal brain scan. However, if all of a sudden a noise occurs in the room, your attention is going to be diverted to that. Your mind is going to be aware that a noise has happened. Or maybe you're in a dark room and all of a sudden bright lights are turned on. And even if your eyes are closed, you can still see that, ooh, the lights have come on, something has happened. What that's going to do is that's going to dip, that's going to suppress the alpha wave. And that's a sign that you are, in a sense, consciously aware of some sort of sensory perception. Something has happened that your brain is now turning its attention to. And that's what the Caltech researchers found. So, you know, I'm not a psychologist. I don't really understand the distinction between what they classify as conscious, unconscious, subconscious, subliminal, etc. But the point is that the brain was aware of some sensory perception when the orientation of the magnetic field was changed. And all other senses were controlled for.
Okay, so just as a follow-up, it sounds as though there was no one who had any distinctive feeling about feeling something different from seeing, hearing, touching, etc. The rest of us can't detect. So there's no distinctive magnetic feeling, so to speak.
That would be a question for the Caltech researchers. I don't know if they asked any follow-up questions on their subjects as to sort of their qualitative experience throughout the experiment, if they noticed the changes. And I suspect the researchers didn't do that because they wanted to have sort of clean quantitative data rather than the sort of more touchy-feely impressions of someone saying, oh, yes, I totally sensed when the magnetic field shifted on me. It wasn't quite that experiment. But I think it points to, there are a lot of experiments. I know Dean Radin was one of the speakers for Noetic Science Institute. A lot of the experiments in parapsychology are like that. They're asking for their behavioral experiments that ask humans to sort of talk about their perceptions. And you can see that sometimes that can be a harder experiment to control for. Whereas when you're actually taking data from EEGs of what the brain is actually sensing, what sensory activity is actually happening, that can perhaps be a more accurate way of discerning what the human brain is perceiving rather than asking people.
Sure, I understand. Thank you very much.
Sure.
This is just a quick one. Jim Barron here again, the chair. Is it your impression that the information from these sensors is transmitted through the optic nerve to the brains of the birds?
Yes, for the chemical compass model that Dr. Ritz originally proposed, that would be the case because the compass itself is occurring in a protein located within the cone cells within the retina of the eye. And within that protein, the cryptochrome protein, there's a specific molecule, this flavin molecule, that is light sensitive. It's a blue-green light receptor. And it's within that light receptor that this molecule has the ability to form magnetically sensitive radical pairs with other molecules. And so that's where we theorize the magnetic sensitivity arises from.
Thank you. And thank you for your presentation. I think for those who are still here, we're going to watch the video submitted by our seventh presenter, Mac Davis. I'll give a brief introduction. Mr. Davis is with Minicircle, and his title is Toxoplasmosis, Endemic Pathogenic Virins and the Unacknowledged Psychospiritual Correlates of Parasitic Load.
This is your last chance. After this, there is no turning back. You take the blue pill, the story ends. You wake up in your bed and believe whatever you want to. You take the red pill, you stay in Wonderland, and I show you how deep the rabbit hole goes. Remember, all I'm offering is the truth, nothing more.
In this presentation, you're about to learn how endemic, latent, infected parasites suppress global consciousness in humans. A little background about me. My name's Mac Davis. I founded MiniCircle, which is a gene therapy research company. We also have a gene therapy clinic in the Caribbean that is doing clinical trials this summer in HIV and in aging. What is a latent infection? It's basically one where the first time you're infected, you may experience symptoms, and then it becomes a very low-grade chronic infection for the rest of your life until perhaps the end. Now, I would argue that the term latent is sort of a misnomer because latent infections, all of these types of pathogens and parasites, are traditionally regarded by medicine as being insignificant, as having negligible effects, when that is absolutely not the case. And these chronic latent infections are having constant suppressive effects, both on physiological health and on mental health. The pathogens discussed in this talk are endemic. That means they are found in virtually all humans. And in many cases, they have evaded testing. Now, I'm going to run through a few different sources to indicate that these diseases are a big deal. You know, cytomegalovirus, almost 100% of people have it in some countries, and it is the most significant viral cause of birth defects. It decreases IQ in children, and it appears to lead to increased morbidity and general health decline in life. HSV is herpes, and the traditional take by medicine is that it is mostly insignificant and that latent infections are common in pretty much everyone and it doesn't matter. However, herpes is the leading cause of infectious blindness in the world. And herpes viruses, when injected into quails, cause severe atherosclerosis and tumors. This study also found that 90% of people with atherosclerosis were positive for cytomegalovirus, which is a type of herpes virus. And only 53 of people without atherosclerosis, only 53% of those people were positive for CMV. So that's a pretty reasonably strong correlation. This is Toxoplasma gondii. This is a single-celled protozoan parasite that is somewhat closely related to malaria, for reference, the malaria parasite. And the way that it works is it's typically transmitted through the mouth. Toxoplasma has succeeded in infecting many of the animals in this world, including and especially livestock. And it creates these cysts full of toxoplasma inside of the muscle tissue. And when this muscle tissue is consumed as meat by humans, if it is not cooked well done, then those cysts can break open inside your stomach and basically use the conoid tip to burrow through the tissue of the stomach and find leukocytes, white blood cells, in order to infect. Once it gets inside of the leukocytes, it begins to replicate and it creates and secretes proteins that make the leukocyte more likely to end up inside of the brain. And once toxoplasma gets into the brain, it starts creating cytokine cascades. It starts creating inflammatory cascades that increase the likelihood of more white blood cells to penetrate into the blood-brain barrier. It uses the white blood cells as a Trojan horse in order to get into the brain. And then once it's in the brain, that's where it really sets up shop. In the brain, it is known to epigenetically remodel the CNS. It controls histone, lysine, acetylation, arginine vasopressin metabolism. Toxoplasmic cysts are shown in mice to control dopamine, GABA, glutamate, serotonin, norepinephrine production and secretion across the entire CNS. The parasite contains the genes to synthesize serotonin and secrete it into the blood, into the brain tissue, in order to create conditions that enhance its own replicative likelihood of success. Now, toxoplasma is very sneaky, as is the case with all of these latent pathogens. It has a way of getting around detection by the immune system, and oftentimes detection by molecular biology diagnostics. Now, in toxo, what happens is it has two different forms. It has the tachyzoid form, which is fast replicating. It's the form that is active in the stomach, gets into the white blood cells, goes to the brain and replicates. And then the bradyzoid form is the long-term cyst form that is known as the latent form. Although it's called latent, it is known to engage in remodeling and neurotransmitter secretion in the brain. So the bradyzoite cysts are full of a lot of toxoplasma cells, and those cells generally have a slower metabolism, and the cysts, the bradyzoites, can transition back into tachyzoites in conditions of low inflammation. So the tachyzoid form causes acute infection, which is very dangerous, and the bradyzoid form causes latent infection, which is also very dangerous. Part of the issue with toxoplasma diagnostics is that there are widely available diagnostics for the tachyzoid form, whether those are antibody or PCR based or antigen based in some way. It's easy to test for tachyzoites. However, that's the most rare form. Like everyone has bradyzoites in them for the most part. And those bradyzoites, there is no commercially available diagnostic for testing for bradyzoites. This is Jaroslav Flagard. I met him in Prague at Charles University last year, and he sat me down. He's the world expert in the sociological and neuropsychiatric effects of toxoplasma latent infection. And he sat me down and told me that he has recently determined, he estimates that 23% of the global disease burden is attributable to inflammatory dysregulation caused by toxoplasma infection. He also maintained that toxoplasma contributes to or causes as many as 1 million fatal car accidents per year. Flager's done a lot of studies on car accident victims and found both that motorcyclists were much more likely to have high amounts of toxoplasma in their bodies. And he also found that people who died from car accidents and were determined to be responsible for the car accidents had much higher incidences of toxoplasma infection. Now, the toxoplasma is believed to decrease reaction time and increase a person's risk appetite. This is the paper where he mentions 23% disease burden estimation in Europe. Table 2 on the right has examples of all of the different diseases whose incidence is correlated with a higher Toxoplasma latent infection rate. There is really a laundry list of different diseases, everything from schizophrenia to epilepsy to bipolarism, depression, anxiety, cardiovascular disease. Really, there's just so many correlations. Some research suggests that autism spectrum disorders may be caused by toxoplasmotic infection. Evidence suggests that toxo may cause brain cancer. This paper notes that it decreases apoptosis and it increases inflammation, and those are both favorable conditions for causing cancer. Okay, now here's where it gets really crazy. I believe that these pathogens have effects on political psychology. So there is a line of research called the parasite stress hypothesis or parasite stress theory, where researchers have found that in populations where there are higher amounts of parasites living in the body, people on political compass score tests tend to lean more towards authoritarianism as their preferred political system. So, what this could suggest is that parasites inside the body incentivize you to desire parasites outside of the body. Now, you may say that this logical leap is a bit too far, but I submit to you that Toxoplasma, when it's in mice, remodels the mouse's nervous system in such a way that mice become sexually attracted to the smell of cat pee. This is a complete inversion of what mice should be most afraid of. I argue that Toxoplasma, when it's in the human brain, is doing the same thing. It does not know that it is not in a mouse brain, and it is inverting people's fear of predators. It is making people love predators. It's making people attracted to predators. It makes people vote for predators. A friend of mine attended the Neuralink launch announcement event hosted by Elon Musk. And after the presentation, my friend asked Elon what he thought about toxoplasmosis. And Elon said, Toxoplasma runs the world.
And Toxo's evolutionary challenge at that point is to figure out how to get rodents inside cats' stomachs. Now, it could have done this in really unsubtle ways, cripple the rodent or some such thing. Toxo instead has developed this amazing capacity to alter innate behavior in rodents. You take a lab rat who is 5,000 generations into being a lab rat since an ancestor actually ran around in the real world, and you put some cat urine in one corner of their cage, and they're going to move to the other side. Completely innate hardwired aversion to the smell of cats, the cat pheromones. And you take a toxo-infected rodent, and they're no longer afraid of the smell of cats, and in fact, they become attracted to it. The most damn amazing thing you can ever see. Toxo knows how to make cat urine smell attractive to rats. And rats go and check it out, and that rat is now much more likely to wind up in the cat's stomach. So this was reported by a group in the UK about half a dozen years ago, toxo behavior people in this totally cool phenomenon. And, not a whole lot was known about what toxin was doing in the brain. So ever since, part of my lab has been trying to figure out what's the neurobiology of this. So the first thing is, it's for real. The rodents, rats, mice really do become attracted to cat urine when they've been infected with toxo. And you would say, okay, well, this is a rodent doing just all sorts of screwy stuff because it's got this parasite turning its brain into Swiss cheese or something. It's just non-specific behavioral chaos. But no, these are incredibly normal animals. Their olfaction is normal. Their social behavior is normal. Their learning and memory is normal, all of that. So this is not just a generically screwy animal. So you say, okay, well, it's not that, but toxo seemingly knows how to destroy sort of fear and anxiety circuits. But it's not that either, because these are rats who are still innately afraid of bright lights. They're nocturnal animals. They're afraid of big open spaces. You can condition them to be afraid of novel things. The system works perfectly well there. Somehow toxo can laser out this fear pathway, this aversion to predator odors.
Unfortunately, most of them do not have available therapies. HPV is the only one that has a pretty effective therapy, and still that vaccine, if you're already infected, will not completely get rid of your HPV infection. There's one herpes vaccine approved in Russia. I am not sure how effective it really is. And the only intervention for Toxoplasma latent infections is designed by a Dr. Uwe in Germany. And what he's been doing is been giving people folinic acid plus Daraprim, the famous drug that Martin Shkreli purchased the patent for and increased the price of to insurance companies. And those two drugs are combined with some type of antibiotic that is determined to have a strong effect on reducing and suppressing toxoplasma populations in the brain. Ought to be. These diseases have been sidelined. People have assumed for far too long that they are not priorities. I would argue that the overall quality of life adjusted risk of all of these latent infections is much more serious than that of COVID. And if a disease like herpes or toxoplasma suddenly appeared and hadn't already been around for all of the human species' evolution, people would be freaking out just the same way that they were about COVID. But because these diseases are already inside of us and have always been here, we don't regard it as a new threat, even though it is a real threat. So the human panic sensors are not going off, but they should be going off. People are inundated into believing that having all of these diseases is perfectly normal. And it is normal. It is normal, but it's anti-health, it's anti-life, and ultimately anti-consciousness. These parasites have infected the human body mind and selfishly use our energy for its own replication. I believe this ultimately causes enhanced aging. Now, one may argue from a terrain type mindset or a permaculture type of mindset that these viruses are natural and we have evolved with them and that they are there for a reason, and that the holistic take of what the human organism is would include these viruses as being an integral part of the whole. And I would argue that that's a very nice perspective to have, and it's very helpful in general. But in the case of these viruses, there are no positive outcomes associated with them. And toxoplasma as well is very strongly negative. So my question for the world is, what would the world be like without these parasites? Our enemy is not the Republicans or the Democrats or the Russians or the Ukrainians. Our common enemy as humans is really these latent diseases that are embedded in our bodies and need to be extricated. We are held in bondage by these diseases. We are given anxiety, depression, schizophrenia, bipolarism by these diseases. And these diseases also affect the way that we make our ethical and political decisions.
The matrix is everywhere. It is all around us. Even now, in this very room, you can see it when you look out your window or when you turn on your television. You can feel it when you go to work, when you go to church, when you pay your taxes. It is the world that has been pulled over your eyes to blind you from the truth. What truth? That you are a slave, Neo. Like everyone else, you were born into bondage, born into a prison that you cannot smell or taste or touch. A prison for your mind.